Bismillah Rahman Rahim, November 24, 9700, paper 2-2, and this is the first video which we are going to discuss. As you all know, this paper is for 1 hour and 15 minutes, and you should first do the questions you're absolutely confident about, and then come back and do the ones which you're not so confident about. The trick to do these questions is that first you please do the paper yourself, and please do it in writing. Do it on a piece of paper or a copy or something, and then go through this video. So don't go through this video first without doing the paper yourself first. That is the rule that I expect you to follow. The olive plant, Olea europea, is grown in many parts of the world. The fruits of the plant, olives, and the oil that can be obtained from the fruits, olive oil, provides food for humans. Triglycerides are the main type of lipid in olive oil. They are synthesized in the olive plant from glycerol and fatty acids. Scientists can analyze samples of different olive oils to identify the fatty acids used to synthesize triglycerides. Triglyceride means one glycerol and to which three fatty acid chains are attached. And the composition of the different triglycerides present. So one, they analyze the fatty acids used and the composition of the different triglycerides. Table 1.1 shows some details of the 5 most common fatty acids found in samples of olive oil produced by olive plants grown in different regions in Portugal. Table 1.1, CD. C ratio D is the number of carbon atoms to the number of double bonds in the hydrocarbon chain. And X is the missing detail. Now you need to look at this very carefully. It's not an easy question. Table 1.1 shows the CD values for oleic acid. In table 1.1, write the values for D in each of the 4 other fatty acids. So we have to write something here, and something here. But this one has been done for us. This is 18:1. And the ratio is C to D is what ratio? Number of carbon atoms to the number of double bonds. Now you can see this is one double bond here. So 18:1, right? So you have to write down in table 1, write the values for D. So what are the values for D? Now in palmitic acid, here, let's look at it here. This is now in this case, in palmitic acid, it is 0. There is no double bond here. And then in linoleic acid, you can see there's a double bond here and there's a double bond here. So this is 2. And then in stearic acid, 0. And then palmitoleic acid, you can see there is 1. So there is this one double bond. All this got you 1 mark. If you got all these correct, you got 1 mark. Then it says in the first column of table 1.1, draw a circle around each of the fatty acids that can be described as saturated. Now that can only be, because this one didn't have any double bond, it can only be palmitic acid and stearic acid. So only these two. It says draw a circle around each of the fatty acids that can be described as saturated. So circle drawn around palmitic acid and around stearic acid. You see, both of these did not have any double bonds. So when they didn't have any double bonds, it means these were the two that you had to circle. There are no double bonds here. There is a double bond here. There is a double bond here. There is a double bond here. And there is a double bond here. Then, of course, if you've done this part of the question before, only then you should see this video. Number 3, state the detail of chemical structure represented by X which is missing from table 1.1. So what are these X's? It's very easy. It's carboxylic acid group. Or you could have said COOH. Or you could have said carboxylic acid group. Then comes to part 4. The analysis of the triglycerides present in the different samples of olive oil showed there are many different triglycerides present in olive oil. Each olive oil is different in its composition, but the same few triglycerides are present in all olive oils. With reference to table 1.1 and the structure of triglycerides suggest explanations for these observations. What are the observations? There are many different triglycerides. And the next observation is each olive oil is different, but the same few triglycerides are present in all olive oils. Now, it's a very basic question. It's only for 2 marks, and you have to be very sensible in answering this question. Each triglyceride has 3 fatty acids. Fatty acids can be saturated or unsaturated. Fatty acids can have different lengths of fatty acid. Number of carbon atoms can be different. So there are about 9 mark scheme points, but I am not going to go into all the details of it. I'm going to come up with the very common thing which everybody should have thought of. Now, of course, there are points like particular fatty acid can be located at a different position in a triglyceride. Different environment conditions of plants grow in different regions. Colder conditions and oils will have more triglyceride with high proportion of unsaturated fatty acids. Then the fact that shared triglycerides have structures suited as energy store. You see, fat is an energy store. And maybe the ease of production or the synthesis is easier in certain plants, in certain olive plants, and not in the other plants. And shared triglycerides composed of the fatty acid present in the highest proportions. So a lot of answers, but the basic 3 simple ones I would only talk about. They only 2 marks for it. So you have to just tell me a little bit about triglycerides. So each triglyceride has 3 fatty acids. Everybody knew this, but I know nobody would write it. Then coming to the B part of the question, glycerol is soluble in water, triglycerides are insoluble in water. Explain why water is a good solvent for some substances such as glycerol, but is a poor solvent for substances such as triglyceride. Now this is a question on the water chapter. What is it? Water. What is a water? Water is a dipolar molecule. Is a polar molecule or a polar or dipolar. Even if you said water is a dipole, I would give you a mark for that. Then water can form hydrogen bonds with polar substances or with glycerol. Water is a polar molecule. It's got a charge on it. Oxygen has a partial negative and hydrogens have a partial positive charge. Water can form hydrogen bonds with polar substances. Sodium, potassium, calcium. That's why ions dissolve in water. Glucose is polar. Glycerol is a polar molecule, so it interacts with the water molecules. So 2 marks, and you had to give me why water is a good solvent for polar molecules. So it was a very basic, but I'm sure some of you didn't know it. Some of you who knew it probably didn't know how to word it. But I mean, I don't agree with this. When children say, Miss, I know it, but I don't know how to word it, that means you just don't know it. Either you know it and you can word it, or you don't know it. If you don't know the wordings of it, then when were you studying? How were you not focused on the wordings? I want to say something to anybody. I want to say, oh, please, can you get me a cup of tea? What do you mean I don't know how to say? So I'm just going to say tea, tea, tea. What is he going to say? Tea, what tea? Tea for telephone. Tea for tiger. I want a cup of tea. So I mean, all these situations that Gen Z is coming up with, I'm sorry, I don't agree with this. I do not believe that you know your subject content. If you know your subject content, you can always write the answer. But actually, you don't know your subject content. And that's why you keep on asking me questions like, how can I finish the course in 2 months? Well, you can't finish the course in 2 months. You're going to flunk it, definitely. Don't try to be, think you can do things in which children did it in 3 years. Now you think you can do it in 2 months. Nobody's going to be able to do it in 2 months. So the wordings of it, water is a polar molecule, water can form hydrogen bonds with polar substances, could be glycerol, and glycerol interacts with the water molecules, or you can say they're hydrophilic. So you could have written all this. Then coming to part C of the question, phloem is the plant tissue responsible for the transport of organic substances such as fatty acids from one area of plant to another. The tissue is composed of more than one type of cell. Name the type of cell that forms the transport vessel of phloem tissue. Sieve tube element. Sieve tube element. Nothing else. Sieve element. Yeah, you will get a mark for that as well. You see, if it's underlined in the mark scheme, that means that is what you have to write. You could not write anything else. If it's underlined, that is the only word we are looking for. They've allowed sieve element as well. Sieve tube element. Phloem was given in the question, so writing phloem is not going to get you a mark. You must be hyper changad if you are writing phloem. It's written in the question, form the transport vessel of phloem tissue. Sorry, Gen Z, you need to be smarter than what you are behaving like these days. Now coming to question number 2, people who become infected with HIV are at a risk of developing HIV/AIDS, particularly if antiretroviral therapy is not available. And now antiretroviral therapy is ART. In people infected with HIV, the use of ART, antiretroviral therapy, also helps to reduce transmission of the virus to uninfected people. Outline 2 control methods other than ART that can be used to reduce the transmission of HIV. There's a whole list of them. Practicing safe sex, screening blood before transfusions, then treating the donated blood to inactivate the virus. If it is in the context of drug abusers, then you say avoid sharing needles, avoid breastfeeding in women who are suffering from AIDS, and using sterile equipment for tattooing or surgery or dental procedures. So any of these two would have got you your 2 out of 2. So this wasn't a very difficult part. I'm sure all of you know it. 2 control methods. It asks for 2 control methods. So I've just given you 3 of the points, but I have talked about the other points which were also available to you. So you could have also talked of all those other points. Any of those 2 would have got you your 2 out of 2. In people with HIV/AIDS, a serious lung disease, Pneumocystis pneumonia, can result from infection of an opportunistic pathogen known as Pneumocystis jirovecii. Figure 2.1 shows P. jirovecii cells in one stage of their life cycle as seen using a light microscope at a magnification of x600. And this is 0.4 micrometer in diameter. Now it says, let's look at the question, define magnification. Now simply define, it's in the syllabus, you must know all the definitions. The number of times larger an image is than the actual. The number of times larger an image is than the actual. That's all. That's all what you have to give me for 1 mark. How many times is this picture bigger than the actual? I tell you to draw your phone twice the size. So you measure the phone and then you draw it twice the size. And so it's magnified 2 times. So define magnification. The number of times larger an image is than the actual. Then coming to part C, figure 2.1 shows P. jirovecii is a unicellular organism. Although the cells of many species of bacteria are the same size as those of P. jirovecii, research concluded that the organism is a eukaryote and is not a bacterium. In 1988, analysis of ribosomal RNA resulted in P. jirovecii being classified as a fungus. Studies of the structure of P. jirovecii have identified that the cell wall is made of polysaccharides such as chitin and 1,3-beta-D-glucan. Explain why this feature helps scientists to confirm that P. jirovecii is not a bacterium. Basically because bacteria have cell walls made of peptidoglycan. The fact that the cell wall is made up of chitin proves that it's not peptidoglycan. So if it's not peptidoglycan, it cannot be a bacteria. Because chitin, and some of you say chitin, it's not chitin, it's chitin. Chitin is not found in the bacterial cell walls. So for 1 mark, you had to say the bacteria have cell walls made up of peptidoglycan, or you can use the word murein, murein. Or you could have said bacteria because chitin is not found in bacterial cell walls. Number two, scientists have identified other features of the cell structure of P. jirovecii. Some of these are listed in table 2.1. Complete each row of table 2.1 so the table shows 4 structural features identified in P. jirovecii, one function for each structural feature, whether the structural feature present or absent in bacterial cells. Present or absent in bacteria. So ribosomes, protein synthesis, yes, they are present in bacterial cells. Then smooth endoplasmic reticulum is for lipid synthesis. It is for lipid synthesis and it is not present in bacteria. Then Golgi body, modification of proteins and lipids, and it is not present in bacteria. Then aerobic respiration is mitochondria. Fungus has mitochondria in it. Fungus is a eukaryote. So mitochondria is present, but it is not, bacteria does not have any mitochondria. Please remember that. I don't know, but some of you keep on forgetting that. Bacteria does not have any mitochondria in it. Now coming to part D, P. jirovecii can adhere to squamous epithelial cells of the alveoli. So this is the alveoli and it has these squamous epithelial cells which are lining the alveoli, and maybe in some you can see the so now the P. jirovecii, which is a fungus, can stick to this. So this is the P. jirovecii or whatever the spelling is, and to the network. So the P. jirovecii can adhere to squamous epithelial cells of the alveoli and to the network of fibrous protein that support the alveolar wall known as the extracellular matrix. Examples of proteins in the extracellular matrix are elastin and collagen. Adhesion, which is attachment of P. jirovecii to alveolar epithelial cells and the ECM, stimulates the growth of its population. So this then starts to divide. Cell surface glycoproteins known as GPA glycoproteins are essential in allowing P. jirovecii cells to adhere to alveolar epithelial cells and ECM proteins. Suggest means give me a possible example or give me a possible explanation how a GPA glycoprotein is able to adhere to alveolar epithelial cells and ECM proteins. It's very easy. GPA glycoproteins binds to a receptor. So on the surface of this, there will be some sort of receptors to which the P. jirovecii attaches. So the GPA binds to a receptor protein on the surface of the alveolar cells, and the GPA is complementary shape. The GPA is complementary shape to the receptor on the alveolar cell or the ECM. So that is the basic understanding which you must. Or GPN proteins on the surface, so suggestion of adhering by attractive charges, or name a bond which could have been formed. So for 2 marks you have to give me this explanation. So GPA binds to receptor on alveolar cells and GPA is a complementary shape to the receptor. This is, or you can have also said GPA acts as a ligand. So you could have also said GPA acts as a ligand. Okay, next part 2. One consequence of the pneumonia that results from P. jirovecii infection is a decrease in the quantity of oxygen that is delivered to body tissues. Explain why a severe P. jirovecii infection results in a decrease in the quantity of oxygen that is delivered to body tissues. Now they've told you this, that it is going to cause a decrease in the quantity of oxygen. Now what could be the reasons for it? So the alveolar cells are surrounded by jirovecii cells. So you've got the alveolar cells. This is an alveolus, right? And you've got alveolar air inside. So they've got the P. jirovecii cell surrounding it. So now the oxygen has to diffuse across a whole lot of stuff. So alveolar cells are surrounded by the P. jirovecii cells. Diffusion of oxygen is decreased. because the oxygen has to diffuse from here into the blood capillary which is surrounding it. And inside this we've got the red blood cell. So diffusion of oxygen is decreased. So this is decreased between the alveolar space and the red blood cell. This is the alveolar space and the red blood cell. Less oxyhemoglobin formed. And alveolar air is not refreshed because elastic fibers do not stretch and recoil, they are impaired. So the elastic fibers do not stretch and recoil, alveolar air not refreshed, so there is a decreased diffusion gradient. So there are 3 marks. You could have written a whole lot of stuff for this. So alveolar wall surrounded by P jirovecii. I've written PJ, but I'm sure you'll have to write the whole of it. P jirovecii cells, or you can say alveolar wall becomes thick. Then diffusion of oxygen is decreased between alveolar space and red blood cells. Less oxyhemoglobin formed, alveolar air not refreshed, and decreased elasticity of the alveoli. Then coming to the last part of this question, P. jirovecii produces an enzyme known as 1,3-beta-D-glucan synthase. The enzyme catalyzes the synthesis of 1,3-beta-D-glucan. The therapeutic drug caspofungin is a non-competitive inhibitor of 1,3-beta-D-glucan synthase. So this is the enzyme, and it is a non-competitive inhibitor. So it attaches to a site other than the active site, and it changes the shape of the active site. So the active site becomes something like this. So the substrate is no longer going to fit. Now the fungus produces an enzyme, this enzyme, beta-glucan synthase. The enzyme catalyzes the synthesis of this glucan. The drug is a non-competitive inhibitor of synthase. So with reference to the mechanism of action of caspofungin, explain how the drug may be used to treat cases of pneumonia caused by B. jirovecii. Now the drug binds to site on the enzyme other than the active site. That got you a mark. Changes the shape of the active site, got you another mark. Substrate cannot bind to active site. Yes, now naturally the substrate cannot bind to the active site because the substrate was something like this. Substrate was something like this. But now the shape of the active site has changed. Now what is the shape of the active site has become this. So this is not going to fit in there. Substrate cannot bind to the active site. So no products will be synthesized. No products will be synthesized because if the substrate does not fit, no products will be made. The cell wall will be weakened, and lysis of the P. jirovecii or the P of the fungus will take place. So bursting, or you can say cytolysis, and this will of course reduce or prevent the growth, population growth will decrease. So basically you had to concentrate on this word, non-competitive inhibitor. And you just had to give me, this is a chapter on the enzymes, which you had to give me ideas from that. What is a non-competitive inhibitor? So you had to give me all that, and this was for 5 marks. So you had to give me at least 5 or 6 points so that if any of the wrong, you get your 5 out of 5. Now you could have said enzyme substrate complex not formed, or you could have said substrate cannot bind to the active site, or you could have said active site no longer complementary to the substrate. Any of those words that you can use. So these were all the points that were possible. Less products produced, cell wall weakened, lysis of the fungus or bursting of the fungus, reduces population growth. Naturally, the fungus should multiply very rapidly. From 100 they should become 200 to 400 to 800. No, but then this is going to stop the population growth of the fungus, and water will enter the cell by osmosis because of a weakened wall, and that will result in, of course, the fungal cell bursting. Question number 3, during transcription a base pairing occurs between nucleotides. Figure 3.1 is a diagram to show complementary base pairing between a DNA nucleotide and an RNA nucleotide. Only the base pair is shown in molecular detail. Explain why 3.1 does not include any phosphodiester bond. Well, of course, phosphodiester bond is between nucleotides. So there is that phosphodiesterase formed between nucleotides. So if we drew another nucleotide here, then it would form a phosphodiester bond with this. So we would have the ribose, and then we would have it continue here. This would be the phosphodiester bond. But we have not shown you. So the phosphodiester bond forms between nucleotides on the same strand. So if we had drawn another one here, then we would have drawn a phosphodiester bond here. Or if we had drawn one here, then we would have drawn a phosphodiester bond here. But we have not shown that. So that is why we do not show it. So phosphate shown only bound to one sugar. Each phosphate needs to be bound to two pentose sugars. Identify and describe the DNA, RNA, nucleotide shown in figure 3. You may add labels and annotations to figure 3.1 if you wish. How do we identify all this? Now this was the phosphate. This is the deoxyribose. This is the ribose, and then this is the phosphate here. Now we know that adenine and guanine are purines, and they are double ring structure. So this is a double ring structure. So it has either got to be adenine or guanine. If it was adenine, then it would pair with uracil, and that only has two bonds in between. But because this is guanine, and there are three hydrogen bonds between guanine and cytosine. So you can see the 1, 2, 3 hydrogen bonds. So double ring base on the left is identified as a purine, and the single ring is a pyrimidine. So you have to just give me all this explanation. So base on the left guanine, right cytosine, dotted line 3 hydrogen bonds, circle is phosphate, pentagon is deoxyribose, double ring on the left is purine, single ring on the right is pyrimidine. So you need to revise your DNA structure and RNA structure if you're not very clear on this page.